Welcome to OncoPharm. I'm your host, John Bazar. I am a professor of pharmacy practice at ETSU's Bill Gatton College of Pharmacy. This episode is sponsored by Kelly C. PharmD, an oncology education company for pharmacists. Today, we're going to talk about some promising early data and some promising late phase data in pancreatic cancer, a disease state that's notoriously just a pain and hard to effectively treat. We're going to start with the modality most likely to make it to market and to change practice in the next, year. I'm trying to read the future. And this is Daraxonrasib, which is a RAS inhibitor, as you might tell by the name, Daraxonrasib. They also were studying Daraxonrasib, and it didn't work because, that's a joke, the way this drug works is targeting RAS in the on or active formulation, and it's a non-covalent inhibitor. It kind of has a pretty cool binding or a mechanism of action. So for a long time, RAS, which is mutated in many, almost probably most solid tumors, if you look at all the solid tumors, RAS mutations are very prevalent in many of them, but it is a very slippery protein. There are not a lot of nooks and crannies. Like the way we inhibit BCR-ABL is there's a nice ATP binding pocket that you can take a small molecule like imatinib, wedge in there, and that prevents ATP from getting in there. It shuts down the BCR-ABL protein. Okay, so you really need a nook or a cranny to have a drug inhibit this. So for a long time, people thought RAS was undruggable, was the quote term, undruggable. And then we had sotorasib, which is a KRAS G12C mutation inhibitor. But there are more than just KRAS G12C mutations. There's G12D is pretty prevalent. So there are other RAS mutations. And what Daraxonrasib does is it binds to this protein, the chaperone protein cyclophilin called CYPA. And binding to CYPA, it kind of forms this new complex that creates a, quote, neomorphic, so new shape, interface that is able to bind to RAS in the on position. The RAS on protein is what's called. So this Daraxonrasib has activity against mutated RAS G mutations, wild type RAS, KRAS, NRAS, HRAS, all the isoforms of RAS. So it has, some people have called it a pan-RAS inhibitor, but it has some different affinities for KRAS and NRAS, etc., and the different G12C, G12D mutations. But it certainly has more opportunity. There are more possible targets with this. And so there's some phase 1 data, but there's a phase 3 study called RASolute 302, of course, RASolute, that the company did a press release last week. 10 days ago, April 13th, about the top-line results of this study, and I think they say they're going to present it at a big national meeting, so it's probably going to be at ASCO later this month, or later this spring, I guess. That would be my expectation. Maybe it'll be later than that. But this is a phase 3 study, and they are randomizing patients who have already received chemotherapy. So these are metastatic pancreatic cancer patients, already received chemo. I think when I looked at the clinicaltrials.gov, it was about 400 or more patients, so a decent-sized study. And they're randomized to either Daraxonrasib or multi-agent chemotherapy, Gem-Abraxane, FOLFIRINOX, liposomal nanoparticle irinotecan with 5-FU, and this is after they've already progressed on chemotherapy, so second-line treatment. And the median overall survival here is 13 months with Daraxonrasib compared to 6.7 months with chemotherapy. 6.7 months is kind of what you might get from gemcitabine alone in the first-line setting. And so you're getting, now in the second-line setting, we're seeing the control arm of chemo with a median OS of 6 months and the experimental median OS of 13 months, which is kind of what you would expect compared to gemcitabine in the first-line setting. So in the second-line setting, for that median OS to still be about the same as what you get in the first-line setting, that's not what you typically see. Typically, the benefit of any drug in the second-line setting is going to be smaller magnitude of benefit in the first-line setting. And this looks to be about the same or even bigger. So really exciting data coming out about Daraxonrasib. And, in the press release from the company, they're like 90% of patients have tumors driven by RAS mutations. They're trying to make a case for the drug. I believe in reading some information about this. Again, we don't have the abstract. This is just the press release. There's some significant supportive care with oral antibiotics potentially. I feel like I saw one place, trying to read the tea leaves here, but something starting to look for in the future, Daraxonrasib. It's always challenging to try to predict how good a drug will be, how common it will be used based on a press release. This is not just a press release that says it met its primary endpoint, which maybe is even progression-free survival, not overall survival. But that magnitude of benefit is really something. Now, what we don't know from this is how many folks in the second-line setting are getting, my guess is most of them are getting Gemabraxane, and probably most got FOLFIRINOX first line. That would be my expectation. We would have to confirm that when we see, the presentation, the abstract, and then ultimately the publication. But certainly one of the more promising new drugs we have seen in pancreatic cancer. So that it's certainly notable. Keep this on your radar, Daraxonrasib. A lot of listeners to the pod tune in because they're new to oncology, trying to get up to speed, or they appreciate quick weekly updates to help keep pace with the onslaught of new information in oncology. If this sounds like you, check out Oncology Insights newsletter, a free weekly email written by fellow OncoPharm Kelly Karlstrom, founder of Kelly C. PharmD and the ELO Collaborative Program. Each week she shares practical pearls, ways to think through oncology concepts without just memorizing guidelines, and insights on staying motivated and growing in your practice. It's written by a pharmacist for pharmacists, and it's easy to read. Just sign up using the link in the show notes. The next possible big advance in pancreatic cancer is much further away. So this is a personalized mRNA vaccine in pancreatic cancer. The methods of this are actually published in May in 2023 in Nature. And so what they're doing is, I'll kind of go through this, and you also see maybe one of the limitations of this approach. Obviously, it's a very technical approach, creating a personalized vaccine for somebody. This was, I think this is Sloan Kettering is where they're doing this. They're going to hate me if I got that wrong. Anyway, it's not here where I practice. But they take 34 patients, they get screened, 32 enrolled, and then 28 are able to get resection of their pancreatic cancer tumor, okay? And then of those 28, they take the cells into the lab and they're sequencing the tumor DNA, comparing to the normal DNA, trying to predict what are the best neoantigens. So these are the new targets outside the cell that you could try to hone the immune system to attack. And then they create autogene cevumeran, which is a neoantigen encoding mRNA lipoplex. So basically an mRNA vaccine. So you have this mRNA vaccine that's you're going to give back to the patient. It is then going to go into cells and create this antigen that your immune system should attack. It's a very similar idea to the mRNA COVID vaccines. And I think this was done in development with BioNTech and Pfizer, who developed one of the first mRNA vaccines for COVID. So that's the idea, right? Is you get their own tumor cells, create an own unique to each patient mRNA vaccine that will stimulate the patient's body's immune system, particularly against these antigens, the same way you would do that for COVID-19, for example. All right. So of those patients, then the plan is, once you get surgery, it gives about 6 weeks to do all this in the protocol to create the patient-specific vaccine. Then you get a dose of atezolizumab, and then you get your autogene cevumeran. So then you get your doses of this vaccine, and then they got 12 cycles of modified FOLFIRINOX. Now this is in the early stage setting, so this is an adjuvant treatment, right? So you do resection of the tumor, full resection of the tumor, and standard would be then adjuvant chemo. They're doing a dose of atezo, probably for priming purposes for our T cells, and then the individualized vaccine, and then modified FOLFIRINOX for 12 cycles afterwards. There's a booster also at the end for 9 total doses of this vaccine. So of the 28 who had their tumor resected, 6 were metastatic before they went on to treatment. 1 was not pancreatic adenocarcinoma. 1 withdrew consent, and then 1 manufacturer fail. Always important to think about. 19 went on to receive atezolizumab, but only 16 received the vaccine. One of those, the 3 who got atezo and didn't get the vaccine, 1 had disease progression, 1 went for different cancer treatment, and 1 didn't have enough antigens. So there are 2 patients that were not able to actually get the vaccine that needed to, okay? So of the 16 that received the vaccine, 15 went on to receive FOLFIRINOX, 1 had disease progression. And so the big news, that's not necessarily new news, although I don't think I've talked about it on the podcast. What is new is that at AARC, this is one of the big, or AACR, this is one of the big kind of basic science oncology research, right? So if you work at at an NCI designated institution, the folks working primarily in the lab, this is their meeting. The ones primarily working in clinic, ASCO is their meeting, okay? So they've got these 16 patients now, and they had assessed 2 had an immune response, and they found 8 of the 16 had a big T-cell response, all right? So of those 8 who had a big T-cell response, 6 of those 8, 6 years later, are still alive, and, quote, most responders remain free of recurrence. So it's kind of a proof in concept, right? But it is encouraging of the folks who got the vaccine, which, if we go back, is 16 out of originally 34 people that were screened, 32 enrolled, 28 got resection. So you see there's some diminishing returns on who's able to get this. Of the 16 that got the vaccine, half had a large immune response. Of the half that had a large immune response, 6 of the 8, 75%, if I'm doing that math correctly, have done very well. Now that's really good, right? And can you scale that up on a larger scale to show benefit? And there's an ongoing phase 2 study looking at this mRNA treatment vaccine for pancreatic cancer. So really interesting stuff. I really hope that the clinical trials go forward with this and this gets done. One of the things that sometimes happen in oncology is you will have these procedures, and this is, you could think of it as a drug, but it's also a procedure. Sometimes these procedures are very technical, and they're hard to reproduce, and you'll have a single center say, hey, we do it this way, and it works really well for us, and we can show you 90% of patients that do it the way we do it do great. But when some other place tries to do it, maybe the survival rates, the success rates are the same as the current standard of care. So it can be challenging with something very technical to reproduce these results, and that should be a hallmark of science, is a reproducibility, and demonstrate beyond, a 5% reasonable doubt, p-value 0.05, that it's better than just adjuvant chemo in folks that get resected pancreatic cancer. So that is what I have for you this week. Some promising stuff in a disease that historically, it's in the metastatic section. It's just chemo. And it's not like fancy chemo, right? So there's hope here. There's some encouraging stuff to follow long term and to think about. And if you have patients that are wanting, the next best thing, that would be kind of what I would be thinking about as far as referral for clinical trials would be this mRNA vaccine or Daraxonrasib. So thank you for listening. You can follow me on me on X at PharmDeepNip and the podcast on other social media platforms at UnclePharmPod. And until I talk to you again, remember, doses matter.